of mitosis would be, so we're going to go life cycle. We talk about this. Let's talk about interphase. We'll just take it that way. What I was trying to get to is I'm actually asking you mitosis, not life cycle of the cell, but mitosis. Prophase is your first phase. Okay? So be very careful on the test. Are we asking you the life cycle of the cell, or are we asking you about mitosis? Because the very first phase of mitosis is prophase. All right?
What's the life cycle?
The life cycle, generally we start with interphase kind of thing.
Okay.
But again, in terms of that, the life cycle of the cell really is the complete circle. You could start anywhere, but mitosis, we actually ask you mitosis, we're asking prophase. Life cycle of the cell, usually it's interphase. Somewhat arbitrary, but there it is. All right, so what does happen during interphase? What are our three subphases? G1S and G2? What is happening during G1? What's up, Jesse? What?
No, I was going to say the DNA replication of the second part of the...
In what phase? No, you're right. Which one? Which subphase?
May I interrupt you?
Oh, yes.
I'm here for the biology 112, and it says it's...
No, we're here. Sorry, that's
Okay.
way back at the beginning of this year. Let me pull that off the wall.
Okay.
Oh, yeah.
Thank you, Ms. Young.
Yes. Welcome. Join us.
Thank you.
Yeah, I was thinking I needed to pull that off. That was way back when we had room problems at the beginning of the quarter, so I didn't realize it was still there. All right. So, mitosis, prophase, right? Was the beginning of that one. We are in G1 of interphase. You were telling me what's going on during G sub 1.
Growth.
Growth. Because what has the cell just done?
It's just started, so it's
It's just undergone mitosis, right? So we have brand new daughter cells, and they are smaller than the parent cell. So they have to reestablish their size. What else do they have to do? Do what? DNA? Not yet. That's our next phase. That's where Jesse was going. Produce proteins and replace any lost or decreased numbers of organelles. Right? All of that makes sense? Maybe. Has it been a while since you've even thought about this? Yes. Surprise! All right. What's happening during the next one, S phase then? There's something going on with DNA. There we go, Jesse's got it. DNA replication, and we will hit that again. So remember to tie all these big concepts together, back together. Remember now we did DNA replication in Unit 8. So then we can talk about unwinding the DNA and the templates, DNA polymerase coming in, right? All of that. We'll come back to that, but that's all going to happen. You know it all now in detail. So we're going to go on to the next phase, which is G sub 2. All right. What's happening during G sub 2 phase?
The cell prepares to divide.
The cell prepares to divide. You're right, Jesse, but how is it doing that? Protein synthesis. Which proteins is it making? Things needed for? Yes, cell division. All right, and what else does it need lots more of if it's going to create another whole cell? Needs more cell membrane, okay? So it needs lots of that. Everything that it needs to do mitosis has to be created here. There's no going back. All right. Is there a checkpoint here or not before we move on to mitosis? Yes. Where was our other checkpoint? At the end of G1. Yeah. All right. Now we're entering mitosis. So our phase then is going to be prophase. And what's going to happen during prophase?
Change in the nucleus.
Oh, stop it. No reading from notes. Okay, the nuclear envelope begins to break up. Yes. What else happens? Yep. Yes. The centrioles start to separate, creating spindle fibers between them. So we're laying down the infrastructure for the cell to be able to do its chromosomes. put them on and be able to split them. The sister chromatids are there, we've created them. What are they doing? They're changing.
Are they, they've already coiled and foiled.
They're starting to.
They're starting to now.
Yeah,
So
they're
they're
starting to
folding
shrink
and
down,
foiling.
coiling up, getting thicker, and starting to move around, right? Okay. Now, next stage,
Prometaphase.
prometaphase. So it's in transition. Where is it? Where are these chromosomes moving to?
The metaphase plate.
The metaphasal plate. What are they called at this point? Chromosomes.
They're still called chromatids, aren't they?
And are specifically, yeah, you're right. What kind of chromatids?
Sister chromatids.
Sister chromatids. And what holds them together?
The centromere.
The centromere. Very good. All right. So then we have the centromere holding them together. Metaphase. What have they done?
Lined up at the plate.
Lined up at the plate. How many sister chromatids are there in a human cell?
4. 4.
4.
Wow.
2. Oh my God, it's getting worse.
46?
46.
Holy cow.
You're not a fruit fly. You're not a fruit fly. You have more than 4. Yes, 46 sister chromatids because each cell needs to have how many in it when we get done?
And 8's 23.
23?
It's
You're
22.
gonna have? Oh,
Can you
now
repeat
you're
the
mixing your
first
processes. 46 sister chromatids. Each sister chromatid has how many chromosomes?
23.
It's
2. Yeah. So how many actual chromosomes do we have, guys? 46 times 2? 92. How many does each daughter cell get? 46. Yes. Yes. All right. Don't start mixing processes.
Messed up right now.
Okay. So lining up at the metaphasal plate. Do we move on or do we have a checkpoint? Yes, ma'am.
What are checkpoints?
So when we were going through the cell cycle, there were 3 checkpoints. This is where the cell stops to make sure everything's okay. The first one happened where, guys? At the end of G1. So when the cell has first come out of mitosis, this is the most important one. All the cell has to do all of its regular functions and be able to create everything that it had essentially given away, replace stuff, increase in number. So if the cell is not working well here, there's a stopping point and it gets inspected essentially. And if it's not good, it's taken out. Okay, so that's a checkpoint. We do another checkpoint before we actually enter mitosis. And this one is kind of, I always think of it as kind of the checklist before the airplane checklist, before you start flying, before you travel. There's no going back, so you got to make sure you have everything. So the cell stops once again to make sure it has everything. If it doesn't, it continues to make it until it does have everything. Okay? So these are stopping points to somehow make sure that you have the materials, everything's okay, that you're not proceeding without something where it can cause the death of the cell. So, is there a checkpoint here to be decide? Yes, no, yes, no. Good morning. Shake your heads, yes. Yes. Yes, you're right, there is a checkpoint here. Very good. Why is there a checkpoint here at metaphase? What happens right after metaphase? Let's put it that way.
The separation of the sister chromatids.
Okay, the separation of the sister chromatids. What happens if those chromatids do not separate correctly? What would happen to cells? Let's say number 19, chromosome doesn't separate correctly, and we get 3 in one cell and only 1 in the other cell. What are the chances that either of those cells were survived? 0. Yeah, just about 0. So the checkpoint here is to make sure those sister chromatids are attached properly, right? Make sure that they're going to be able to come apart during anaphase. Alrighty?
How do you pronounce the little things that clasp onto the centromeres?
Oh, the
Is it
kinetochores?
Kinetic cores.
Yeah,
I was kinkochores or, yeah, it was really bad.
I know, isn't that terrible? Yes. So built into the centromeres are little attachment points, and those are called the kinetic cores. We didn't get into this, don't freak. This is Kim being thorough. So,
I was just on the picture, I just wanted to make sure.
so what they're gonna do, now what is, besides grabbing hold of the spindle fibers to make sure they separate properly, does anybody, can you make a guess at what else they do? Kinetic? What is,
Energy. Create energy.
if we're talking about kinetic energy, is that energy of stored energy or movement energy?
Movement.
Movement energy, right? Kinesiology is the study of movement. So kinetic cores do what to the chromosomes?
Move.
Move them, yeah. They're the little walking proteins again that are going to walk these guys away from each other during anaphase and get them up towards the poles. Now, you don't have to know that, but if you were curious, now you can sleep well tonight. All right,
I could sleep well.
that's right. I know, that was keeping you awake. I felt it was my duty to let you know. All right, so anaphase, we're moving away from the metaphasal plate. What comes next? Telophase. And what are we going to do during telophase?
Our nuclear envelope starts to come back,
Yes.
and the daughter cells go to, like, one goes to the north and one goes
Right,
to the
we're
south, so
splitting.
we're
We're
separating
cons-
completely.
Yep, and we'll often see cytokinesis happening during telophase and telophase as well. What's going on with the chromosomes?
They're becoming like spaghetti noodles again, like chromine.
Yes, back to chromatin, they're spreading out. The nuclear envelope is being reestablished and the spindle fibers are being broken down. Yeah, it's essentially prophase and reverse, guys, so that we have to kind of put it all back together so it's good to go. Is the cell diploid or haploid at this point? Diploid, haploid, diploid.
They don't become haploid until
You gonna change your mind?
Yeah, and then
Meiosis
when
two,
they
right?
are in it, now they are halfway.
So mitosis, what's the result at the end of mitosis? How many daughter cells?
Two.
Two. Are they diploid or haploid?
Diploid.
Diploid, right? We still have 46 chromosomes, a complete set from each parent. Makes it 2N. Are they genetically diverse or genetically identical? Identical. All right. So that's mitosis. What do humans use that for? Cell growth. Repair. Yep, absolutely. Okay, so our next one is meiosis, and this is a completely different reason that we do this. This is for the production of what? Gametes, eggs and sperm in humans. Yes. So when we get done, what do we want? How many daughter cells should we create? 4. How many chromosomes should they have? Emma says 23. You guys agree?
Yeah, they're haploids.
They're haploid, so they should have 23. And genetically, are they alike or diverse? They are diverse. Okay, so that's our goal. How many divisions do we have?
2.
How many replication events do we have?
2.
1, 2, 6, 24. A lot. We never stop. Don't panic during the test, guys.
Meiosis. we
Deep
have
breath.
1.
How many times does the cell go through interphase? Once. How many times then does it replicate DNA? Once. So if you stop panicking, take a deep breath and think about what you know. You know replication of DNA happens during S phase of interphase, and how many times does the cell go through interphase during meiosis? Once. So it can only replicate once. Okay? So don't always go, I don't know this immediately, and panic. Think, what do I know? Okay? Sometimes we ask you a question from the side and you only recognize it like this, but you know the answer. Okay? So there's more in there than you think. It gets nerve-wracking when you don't see it on a test question. You don't immediately recognize it. Our first thing is to panic, and then once you go into panic mode, then your brain starts shutting down. Okay? So just go, what do I know? Just Write down everything you do know. What do I know about DNA replication? When does it happen? Somewhere along the way, up's gonna pop interphase. All right, and then you look at it and go, well, the cell can only go through interphase once, and it divides twice, so it only replicates once. Okay? So don't panic on those things. That gets you in trouble. All right, so the cell does interphase exactly the same way. What's the first part of meiosis? What's the first stage? Prophase 1. This is a highly significant phase in meiosis. Why is this so very, very important? Yeah, the crossing over. So we're going to do something a little different. The homologous chromosomes are going to find each other. What does that mean, homologous chromosomes? Same? Identical? They carry the same genes. Yes, be careful between being identical and carrying all the same genes in all the same locations. Because identical implies the type of gene, the allelic form of the gene, is the same, and that's not always true. Okay? Because dad could give you brown eyes and mom could give you blue eyes kinds of things. But the eye color location is in the same spot on that same chromosome. Yes?
So you're saying crossing over happens in prophase 1?
Yep. And this is the biggie. So all of your chromosome 1s find each other, mom's chromosomes, sister chromatids find your dad's sister chromatids, and they join up, and then we have the crossing over. When those guys form up, they make a what? Tetrads, yes. And so when you split out, splice and dice, and make essentially new copies, new variations of those chromosomes. Then what happens to the tetrad? Does it split up and go its own way, or hang together?
It hangs together.
Hangs together. What's its next stop?
Metaphase.
Metaphase.
1.
Aha.
Excuse me, sorry.
All right. So what do we know about the difference between the mitosis phases and the meiosis phases? How do you recognize a meiosis phase as an answer? The numbers, right? Don't forget, meiosis has 2 divisions, so every phase is numbered. Mitosis is not, so they're just prophase, metaphase, anaphase, telophase. So that's another one. If it's asking you a mitosis question, don't go for anything numbered. Vice versa, if we're asking you a meiosis question, make sure you do pick a numbered one. All right. So metaphase 1, the tetrad lines up at the metaphasal plate. How many tetrads are there?
Oh.
46, 23. One of those is the correct answer.
I think it's probably 23 because you need to make 46.
If each one, yeah, Sam and Jason are right. There are 23. Remember the tetrads now are grouped together. So there is 4 technically chromosomes there. That makes the 92. It's going to be 23. Okay? So we have 23 tetrads lined up at the metaphasal plate. What separates during anaphase 1? The tetrads, or do the centromeres break?
No, we don't know that.
The tetrads separate.
The tetrads separate, yes. And they go their own way. That separation, anaphase 1. What's the next phase?
Telophase.
Telophase 1,
1 and cytoplasm.
1. When that completes, how many daughter cells do we have?
2.
2. Are they haploid or diploid?
No, they're diploid right now. They don't become haploid until meiosis 2.
Dang.
They are haploid, okay? Now this is the tricky part.
Seriously.
I know it. This is the really messy part. Remember, haploid and diploid is not necessarily number of chromosomes, it's complete sets of chromosomes. Do you have a complete set from mom and a complete set from dad?
No.
One N is a complete set of chromosomes from one parent. So 1N plus 1N makes 2N, that's our diploid? Or do you have a mishmash at this point? Even though there's 46 chromosomes in those cells, they are not complete sets from each parent. That makes them haploid, okay? So most of the time, haploid means half the number of chromosomes, but at this one, it's one of those tricky ones. So they are technically haploid at the end of meiosis 1. 2 daughter cells. Do they go through interphase? No. Everybody agree?
Yes.
Excellent. Next stop then, what's the next cycle?
Meiosis.
Prophase 2. Now the rest of this, meiosis 2, looks very much like what other process? Mitosis. But now we're down to how many sister chromatids?
Not at the end.
It's that
Humans
picture.
don't need all that genetic material, let's just get rid of it. 23.
Oh.
Okay? Each cell got 23 sister chromatids, so unlike mitosis, where we had 46 sister chromatids lining up, now we have 23 sister chromatids lining up. And it looks now, from now on, it looks just like mitosis. Okay? So if you learn mitosis well, and you only have to learn the really first part of meiosis, then you're good to go. Do you have a question? No? Okay. All right, so metaphase 1, line up on the metaphasal plate. What breaks during metaphase 2, sister chromatids are lining up on the metaphasal plate. Anaphase 2, what breaks? The centromere breaks at this point. Yep. So we finally break the centromere. Chromosomes move away from each other. Prophase, looks like prophase in reverse, telophase 2. When we are done, what's our end result? How many daughter cells?
4.
4, there it is, finally, 4. There's your 4. All right, are they genetically identical?
No.
No. Are they haploid or diploid? Haploid. What was the big major events that happened here that didn't happen in mitosis? The crossing over and the tetrads. Yep, that's what stands out significantly on these guys. All right. So then take a look. Don't forget things like non-disjunction. Make sure you know those terms and things like trisomy 21, Down syndrome, and then the the karyotype problems that we had, mostly with the 23rd chromosome pair. Okay, so things like Klinefelters, Turners. Just know which ones are autosomal and which ones are sex chromosome related. All right, good to go? And let's talk about Unit 7, tying right into meiosis, which is, of course, the genetics unit. All right. So again, knowing what dominant and recessive means. Dominant doesn't always mean the most prevalent form in a society or in a population. Okay, dominant means what? By true biological terms, if there's a dominant gene, it will do what? It'll stand out. It will express if it's there. That's what dominant means. It does not mean that it's going to be the most common. All right. So what are our terms if we have 2 dominant genes? Homozygous. Dominant, if we have a dominant and a recessive. Heterozygous. And 2 recessives. Homozygous recessive. All right. Make sure that you know which of your diseases are autosomal and which ones are sex-linked. It's easier to know the sex-linked ones, right? So which were our sex-linked disorders that we did?
Klinefelters and Turners.
No, now you're talking karyotypes.
Sorry.
Things that are riding on the X chromosome mostly. Hemophilia. Hemophilia is sex-linked.
Colorblind.
And color blindness. Yeah. So it's a lot easier to remember those two are sex-linked. Everything else should be autosomal. All right?
So it's
Sickle cell is autosomal.
hemophilia.
So,
and
yes.
colorblind.
Yep. You threw me off with that one, Jesse. Is it? I don't know. I might be absolutely right. All right. Can I go like chalk? All right. So if we have an albino hemophiliac male, kind of in bad shape, what would his genotype look like? What's genotype? Your genetic makeup. How many letters do you have for each trait in your genotype?
2.
Yes, I'm very sure about that.
2.
Right? You show it as a diploid state, so you have to have 2 letters for each. Is albinoism dominant or recessive?
It's a recessive.
Recessive. So for our albino hemophiliac male, what is his genotype?
Well,
So does he have big A's or little A's?
the little A's.
Okay, so 2 little A's. And hemophilia? Okay, so it's sex-linked, X little h. It's a recessive disorder. And a 1. All right, he marries a woman who is normally pigmented, but her father was hemophiliac. No, her father was albino. And her mother, now let's just make her normal. Okay, no problem with hemophilia. She's homozygous dominant for it. All right. So what's mom's genotype?
She is a
Normally pigmented means she has to have at least one what?
dominant.
Big A. She's got to have this. The question is, what's here? Now, if her dad was an albino, she could only get a little a from him, right? So she would have a little a. And if she's normal homozygous dominant for hemophilia, X, big H. Okay. So, what kinds of gametes can each of these guys make?
Big A, little A.
Okay, so dad can only make a little a,
Oh.
X, with a
Little A.
little h, and little a with a y. What about mom? Now, and only now do I know how big my Punnett square is going to be. You cannot skip this step, or you'll never get it right. So how big is my Punnett square? 4. 2 by 2. White chalk, sorry, I found some over there. Without it being kindergarten chalk. Okay, now I always just put my mom's across the top so I can keep track of them. My dad's down the side. And I check. Seriously, guys, I check everything before I move on. Little mistake, it's like math. You get the big stuff, but the little stuff will mess you up, and you get the wrong answer. It's going to hose you for the test. All right.
Big A, little A.
Big A, little a.
X, big H. X, little
Okay.
A. Little A, little A. X, big H. X, little H. Big A, little A. X, big H, Y. Little A, little A. Same thing.
by adding in sex-linked chromosomes, we actually end up with 3 pieces of information here. We look at, for each person now, we look at skin coloration, okay? We also look at whether or not they're hemophiliac carriers or so forth, normal, and also gender, all right? And don't forget in your summary that you have to analyze the whole person. You can't say half are girls, half are boys, half have hemophilia, or half have albinoism and half don't. You have to say who has what, okay? So make sure it's a summary altogether. So we have, under the females, we can split them up this way. We're going to have one who has normal pigmentation and carrier, one who is albino and carrier. All right, males. Normal pigmentation and normal heme. Albino and normal heme. Yep. What is our ratio? 1 to 1 to 1 to 1. Yep. Okay. So I have on my Moodle website, and for those of you guys who are visiting from Luke's, if you guys email me, I can put you on guest for my Moodle website. If you want to access, there's some really great websites on the left side for lecture resources. There's the Arizona Biology website that we did in lab for doing the mitosis, looking at the onion root tip slides. That's a really nice one. Takes you through step by step, and it asks you, it's like multiple choice. If you pick the wrong answer, it tells you why. Why did you get it wrong? No, you missed this step kind of thing. So that's a nice one to do. There's a tip sheet there as well. And another one that I found that has some nice linkages, problems, and then links to more problems like that. Again, pay attention to what we did in lecture and lab. We're not, if we didn't go that far in certain ones, don't worry about them. Some of these websites are going to go pretty in depth. If we didn't go there, don't worry about it, okay? Use your lecture objectives as a guide for that. All right? So are you guys comfortable fairly with that? These really are practice, practice, practice. The more you do the steps, the more careful you are, the better that these will go. Most of the time, you guys get it like 90%, but when you go to work it, you've missed a little something, and then that messes you up. Like I say, they're a lot like math. There's not a lot of give in this.
Are we going to be having another genotype?
In what sense?
Like, are they going to give us the letters that we can?
No, we will not give you the letters. So you should know which things are basically of the autosomal stuff, what's dominant, what's recessive, those types of things that we've done, and know which ones, again, are sex-linked. Hemophilia, colorblindness would be the only two I think that we would ask you on. Pedigrees, you guys remember your pedigrees as well? Okay, so tracking that through. Yes, Kim?
I just had a question about as far as the Punnett square, it being 4 versus that larger one where the larger one when we did it on the green sheet review, it was like big B, little b.
Right, because
that's
actually,
for.
It depends on how many gametes your parents actually create.
Because this has 4 and the other ones had 4, but then we created. It just seems like when we started this, I wanted to make that big Punnett square.
Okay.
How do you know when to not make the huge Punnett square?
So what I could do is I could take, it works both ways. I could have done little a with X, big H, little a with Y, little a with X, little H, little a with little y, okay? So I could have had 4 different gametes here,
Okay.
okay? And I could have had 4 different types, or 4 gametes here, but I really would have only had 2 types.
Okay.
So what I'm showing you at this point is a shortcut. You only need the different types of gametes that you make. you out, okay? If it truly does, makes you uncomfortable, you can do it the long way, your numbers will come out the same, your answer will be exactly the same,
Okay.
okay? But if you get it, that there's a shortcut and it makes obvious sense to you, oh yeah, well, of course, feel free to use it. But otherwise, you could absolutely have done it with the 4 and 4.
Okay.
Okay? It works out either way. Just make sure if you do it for one parent, you do it for the other. You gotta stick with the method. Either one's fine. Just make sure you're consistent. All right?
I'm just
Yes.
curious, did you start at Unit 6 today?
Yes.
Okay.
Yeah. We'll go briefly back through 1 through 5 at the end.
So is the test mostly Unit 6 on?
So it's half and half.
Okay.
But since we've been through and tested on 1 through 5 units, we're not going to spend a whole lot of time here. But we'll talk about that at the end, which should cover. But yeah, the test is about half and half. 90 multiple choice questions, 2 essays. Each multiple choice worth 1. You'll have like an hour and 50 minutes to complete the test. Okay? All right. Now, back to interphase and yes phase. When we talk about DNA replication. Here's what I want you to know about this chapter. Do not mix these two processes up, and I said this repeatedly in my groups. Remember that DNA replication and protein synthesis are 2 completely different processes. All
Can you say
right?
that again?
DNA replication and protein synthesis are 2 completely different processes. Do not mix them up and do not mix them together. Okay? Entirely different enzymes being used, entirely different outcome. The only thing is that there's DNA involved in both of them. That's it. Okay? This is the biggest mistake that people make on the test. They get panicky, and then it all starts blending together. Like, no, there's a polymerase in there somewhere. Okay? So be aware of this one. This one's a toughie in terms of keeping it separate. So I would draw it out, make a flow chart, whatever it takes so you can drill through and know these separately. Okay? Yes, Jason?
DNA replication and protein synthesis?
Synthesis, 2 entirely separate processes.
Okay.
Yep. All right. So when we talk about DNA replication, this is where we're talking about Unit 6 and our S phase of interphase, DNA replicates, right? And we do this either for mitosis or meiosis. Absolutely. Don't forget the meiosis part of that. Sometimes people do. All right. So what do I have to do if I'm going to replicate DNA? First thing? What?
Unwind the DNA.
Yes, I have to split the helix. Which enzyme splits the helix?
Helicase.
No?
Helix.
Helicase.
Helicase. All right, these guys are actually named nicely, not some silly names that don't make sense, which most of biology is like. But helicase will unwind the helix. All right. It will do that and create little bubbles, origins of replication. Which enzyme moves in next? Polymerase. Which polymerase? DNA, because what are we making? DNA, yes. So DNA polymerase comes in next, and it will read the template parent strand. Which direction does it read it?
From 5 to 3.
3 to 5? 5 to 3.
Okay, our options are 3 to 5 or 5 to 3 that we are reading.
No, it's 5 to 3.
All right, first
You
time.
have
We always build 5 to 3, so we have to read 3 to 5, okay? That means one strand is called the leading strand. What is the other strand called? It's got bits and pieces, so it takes a little time. Can't quite run as fast as the other. Tends to behind. Right? When you're a kid, if you had a younger sibling and you outran them, they were behind. Lagging. Lagging behind. Anybody found that one? Make sense? Not a clue? Did you ever say to anybody, step it up, you're lagging behind? Excellent, thank you. So, if the strand is not being able to be made as fast, it's lagging behind. So it is there's the leading strand and the lagging strand, yes. See, there's a reason for these things. Yes, really. All right, now that lagging strand is being built in fragments. Which enzyme will seal those fragments for you? Ligase, very good, yes. All right. When we are done, how many rings should there be from one side to the other?
There will be 6. 1, 2, 6.
Okay, so let's back it up.
Oh, I totally.
We talk about purines and pyrimidines pairing together. Which of our bases are purines?
The adenine and thymine? No, adenine and
A and G.
A and G, Jason's right. A and G. The pyrimidines,
cytosine and thymine.
C and T. Yes.
Am I saying it?
All right. Which of those has 2 rings?
The pyrimidine.
The purines or the pyrimidines?
Pyrimidine.
Purines, and the pyrimidines have
1.
1. If those 2 join up and create the step between the 2 sides, how many rings will there be from side to side?
3. Are we counting the?
Just the rings and the nitrogenous bases.
Okay, we're not counting the.
Not sugar, no, just the sides. That's our phosphate sugar backbone. 3. Yes. 3. 3 is your number.
So there should be 3 rings.
All right. So 3 nitrogenous base, 2 nitrogenous bases, 3 rings if everything goes okay. We can do DNA repair if not. All right. When you get done, what holds the 2 daughter chromosomes together? Very good. Very good. All right. See if I can fool you on that one. I couldn't. That's great. All right. So, DNA replication. Pretty good on that one. That's pretty short, straightforward. Yeah. Protein synthesis, not so much. All right. So, how much of the DNA opens for protein synthesis? How much do we copy here? The entire side? What is that designation of the directions for a protein called on the DNA? What is that?
Triplet?
Starts with G. What you guys makes them?
James?
Genes. Not with G though. Right? The directions for a protein is called a gene. Yes. All right. So we're only going to open up for that protein that we need. You guys will get into how we do this in A and P, how that signaling goes on. So we open it up. Which enzyme does this? Yes, RNA polymerase, because we are actually making, are we making DNA or RNA?
RNA.
RNA, so we're moving it to the nucleic acid RNA. We're not doing more DNA. So RNA polymerase does the whole thing for us. Makes a copy off of the DNA, but it does it in RNA form. What do we do to the RNA before we send it out to the cytoplasm? What kind of modifications do we make? Tails, heads and tails. Capping, yep, cap the ends.
Introns out.
There you go. Yep, the introns are snipped out and the exons are spliced together. And then that heads out of the nucleus through a, how does it get out of the nucleus?
RNA.
This is way back to unit 3. Well, it's technically now messenger RNA, but how does it get out of this nucleus? What does the nucleus have in its membrane that allows it to move out? Holes, officially known as
Pores.
pores. Yep, the nuclear pores. Okay, that was a blast from the past of cell types, your organelles. Oh, you got to know all those as well again, guys. All right. Here, lots of fun. Lots of work ahead of you. Okay,
So transcription is part of protein synthesis?
it is. Yep, so transcription, translation. Transcription is just making a copy of the nucleic acid from one form to another. So DNA to messenger RNA. Yes. Now we're moving out for translation into the cytoplasm. What do we need to help us do this? What organelle?
Ribosome.
Ribosomes. And the two places we find ribosomes?
Rough ER.
Rough ER and the cytoplasm. All right. So the ribosomes will hook up around the messenger RNA, and the two parts comes together. What is the code for our first amino acid that's coming in? AUG, coding for the start, or what amino acid is that? Do you guys remember? Methionine, MET, methionine. So yeah, our start codon. How many stop codons do we have?
3.
3. All right. So what is going to bring in your amino acid? You're awfully spunky there. Nadia says tRNA. mRNA is what's already hooked into the ribosome. What is going to transfer your amino acids in?
tRNA.
tRNA. Yes, she's right. All right, where are those amino acids coming from? Big picture. Where do you get your amino acids? From food? Yep. You can make most of them. How many can you not make? This is real trivia. You don't have to know this for the test. Do not panic because you don't know this. How many do you have to get from food? 20 is the total number. You make 8. Your 8 essential amino acids. You ever hear this for food? Breakfast cereals has your 8 essential amino acids. This is why vegetarians have to be very careful about their protein sources, because you eat a cow, you get all the amino acids you need, but if you don't, if you're eating plant material, you have to vary. All right, so 20 amino acids. Transfer RNA will continue to bring them in. What is the process of joining amino acids together? What's that called? Dehydration synthesis. Yes. Right back to polymers, macromolecules. Unit 2, all the chemistry. All right. What is produced as a byproduct? Water. What kind of bond forms? Covalent, and specifically for the peptide bonds. Yes. All right. When does the amino acid chain, when is it finished?
When there's a stop codon.
When there's a stop codon. Yes. And then what would be its next job?
To release.
To release and
Start coiling.
start folding,
Folding.
right? Then we're back to chapter, or was it chapter 3? 2.
Primary.
Macromolecules? Is that, no, I think 3. I think you're right. Yeah, it's unit 2. Levels of protein folding, okay guys? So we're coming back around to unit or midterm 1 type of material, tying this all together. All right. Will messenger RNA be totally done then and just go get recycled? What will happen to it? Goes again. Ribosomes keep hopping on and make it as long as the signal comes in. When enough protein is made, the signal shuts off. Is that a negative or positive feedback?
Negative.
Are you sure?
Yes.
Yeah. Excellent. Yes, it is. All right. Chapter 12. Viruses, again, just the basics. What are our 2 cycles? What 2 cycles do viruses possibly have? Lysogenic and the lytic cycle. If you catch a cold this weekend, which cycle is that virus using? Lytic cycle. Going to active immediately. If you have HIV but not AIDS, then it's lysogenic. Yes. All right. Plasmids? What are these used for? Okay, yes.
Sorry, can
No,
you
go
go
ahead.
back to the viruses again? We didn't go over those.
The 2 cycles?
Yeah,
or not at
the
all?
cycles, yeah.
So when we look at viruses, there's 2 types of life cycles a virus could possibly have. Some have both, some only have one. The lytic, L-Y, hang on, rewrite it for you. Erase start. Okay. All right. So our lytic cycle, okay, this is where the virus comes in, immediately infects you, takes over all of your cells and starts turning them into virus factories.
Okay.
Okay? Lysogenic are the sneakier ones. These guys come in, get into your DNA, and then wait. Every time you replicate that cell, that viral DNA gets replicated with it. These guys will need an environmental cue, some type of trigger, to go active. They'll then go into the lytic cycle. Yes?
Would that be like tuberculosis then?
That's a bacteria.
Okay.
Yep. So that's a different one. HIV, like I say, this is a lysogenic cycle first. Usually there's some type of environmental trigger. Then it goes into its lytic cycle, and that's when it's called AIDS, acquired immune deficiency syndrome. Okay?
So, in lysogenic, it sits where?
It's in our DNA.
Okay, it sits in the
Yeah, it just inserts itself right into our DNA, and so every time you replicate the cell, you've made another infected cell. All right? Yeah, that's why these guys are nasty. They're like little sleeper units. All right. Anything else? Is that good?
That's great.
Okay. So plasmids, who has these? Let's back that up a little bit. The bacteria have these. And what do we do with these guys for plasmids? Why are we interested in plasmids?
Can't you use those to fight viruses because they're that little moonland looking thing?
That's viruses to fight bacteria. Plasmids, what do the bacteria do with their plasmids? Do you guys remember talking about this? These are the way that they kind of get around their limitations of asexual reproduction. They can pass plasmids back and forth between each other. They make a whole bunch, pass them out to their friends. It's like you guys bootlegging copies of, an album, burning CDs for all your favorite friends. Okay. Only this isn't illegal. This is a great strategy for survival. So this increases diversity among bacteria without having to rely totally on mutation. That means they give away plasmids, pick them up very easily. They're bringing in that genetic information from other bacteria. So what we can do with plasmids then is get them to put them down. What do we do with that plasmid then if we want to have them have the new genetic information that we put there? We're going to we isolate it and insert that gene. That's where we start talking about the sticky ends, guys, right? Clipping at a certain sequence to get it in. What are those enzymes called? Restriction enzymes, yeah. And that allows us to be very, very specific about where we insert that gene that we want those guys to start creating proteins for us. So whatever that sequence is here and here, we clip like this, okay? So that the hydrogen bonding is still present, creating sticky ends. And then the gene that we want to insert into that plasmid has those matching sticky ends. So this is our bare end now of the plasmid. The gene that we want slides right in and hooks in, sticks. Now, it's not great, it's hydrogen bonding, but it holds long enough for what enzyme to seal it up? Ligase. Yeah. So we seal it up with ligase, and now the plasmid has what we want in it, and we can convince the bacteria to pick it back up, and now the bacteria has the gene that we want. And it starts making our proteins for us. What would be an example of that? Insulin, pesticides, yeah. So those are things that we can have the bacteria do for us, okay? That's our genetic engineering. All right? Yes. Any questions? Yeah. Now, of course, we're doing that with lots of food items as well, genetically modifying them. We won't get into the whole discussion of that, but that's getting a little bit scary as well. But that's currently what we do. Again, restriction enzymes cut it at a certain point. DNA ligase seals it up, and then the bacteria makes your protein that you need. Okay? Evolution chapter. Okay. So evolution, just essentially modification through descent. What is the driving force of evolution? Natural selection. Yep. Does that increase or decrease the genetic diversity of a population? It increases? Decreases. Yeah. What do we need to increase genetic diversity? Mutations. Yes. So natural selections and mutation are working in opposition to each other. All right. Artificial selection, how does that happen? Who's looking for specific characteristics? Humans. This is where we get involved, right? So, examples of artificial selection? Dogs.
Mustard seed.
Mustard seed, agriculture. Anything else you can think of? That's it? For artificial selection? For evolution is natural selection, environmental pressure. So when we talk about environmental pressure, what you have to remember is that changes with the environment. Natural selection is not the same in every environment. So each organism is adapted as best as possible to that environment. Okay? It's been pressured, it's been pushed to those sets by the environment. And that's working on the material that's already there. You can't create something totally new. Well, now we can, but that's us again interfering. But we can't just, I mean, it's not like you're going to be able to get humans that are totally webbed in 2 generations and make them natural swimmers and meal breathers. Okay? That's not going to happen in a short period of time. All right, what was fitness? Reproduction. So the one who reproduces the most viable offspring is considered the fittest, right? So survival of the fittest obviously makes sense. We're talking about your genes, not necessarily you, but your genes. And of course, the more kids you have and the more kids they have, the more your genes get passed on. All right. Molecular biology, fossils, comparative embryology, comparative anatomy. Make sure you're familiar with those things that we had on our green sheets, which I think were white this that week. But those different types, know examples, be able to classify what those were. The different selection types, this is also true, even when we talk about, doesn't matter if it's artificial selection or natural selection. This is what I saw sometimes when I was grading those labs for this. You guys are like, well, that's artificial selection. Even artificial selection has these different types, guys, okay? So, what was disruptive selection? What were we choosing for?
We have two different.
So we have a bell curve distribution of some trait, okay? Are we choosing for the middle part, the most common, or the extremes? The extremes, yes. How common was that?
It's rare.
Not very. Yeah, it's fairly rare. That's our most rare that we see. Probably because speciation happens a lot faster with that. All right, what about directional selection?
It's for like antibiotic resistant.
So antibiotic resistance, we're only selecting or the environment is selecting when we put all those antibiotics in for the most resistant. Everybody else dies off. The most resistant survive. There's changes. We get even more resistant bacteria being born, if you want to put it that way, being created. And so we keep selecting, or the antibiotic selects for those that are most resistant. So that's environmental selective pressure, but we put it in the environment. We see that in skin color from humans as we moved away from the equator. Selective pressure was for lighter skin, for more vitamin D production. We see that in what we bred for dogs, right? We push for a single trait in dog breeds, or a couple of them, we push them in certain directions,
Yeah.
especially size, right?
Yeah.
We push them one direction and we push them the other. Okay. What about stabilizing? What are some examples of that?
Height.
Height, weights, IQ, birth weights, even skin color, okay? We tend to see that most in the middle, all right? All right.
And that's the most common one, correct?
For
Or
what?
when we're talking about population stabilizing is, I guess, the most common.
Yes, stabilizing is absolutely the most common. Yeah. As long as the environment stays fairly the same, we don't want things to move one way or another because it would not be adaptive. Okay? So as long as the status quo is maintained, we, essentially, we want to be the same as well. We want genetic diversity, but we don't, like I say, if you even just like bone thickness, you wouldn't want to have all of a sudden going to much, much thinner bones in the same gravity, because then bones would start breaking, become maladaptive. All right, mutations, good or bad? Oh, I'm sorry. Yes, ma'am. No, please ask.
So dog traits are directional.
Yep.
And so could you give me an example of disruptive?
So there was the case of the lizards. Did you guys talk about the lizards in New Mexico, Black Sands, New Mexico? Okay. Or White Sands, New Mexico. Okay, so in White Sands, New Mexico, there's this black lava rock that sits on pure white sand. Crazy contrasted environment. And there's these little lizards that live in that area. And they range from black to white. And when we look at where the black lizards can camouflage best, they can do that on the lava rocks, so they hide. The white lizards can hide in the sand and they're good. The gray lizards get eaten. They can't hide anywhere. So what we're seeing is very few gray lizards and lots of black and lots of white, okay? Generally what we see in this type of situation is that probably what we'll see is the white lizards will pretty soon not breed with the black lizards, and essentially we'll have two new species, and there won't be those variations. Or if there are, once in a while something crops up, it'll get eaten and probably removed from the gene pool because it'll be so visible. Okay? Mutations? I'm sorry, yes.
What about peacocks since they stand out so much?
There's a balance there, okay? Between being big and showy and sexy to the ladies, but not so big that you get eaten before you can mate. So that's a stabilizing kind of one. where, you know, bigger is better in terms of sex, but you gotta make it there. So if you get too big of tail feathers, too big so that you can't run away, you're gonna get eaten before you can breed, and that won't reduce that bigness. Okay? So that actually ends up being stabilizing. All right, any other questions? Sexual selection, talked a little bit about that one, which was kind of the peacock one. So the ladies are choosing on that one, influenced by predators as well. Peacock tail feathers. These guys are big, they're showy. That seems to be rather dangerous, and it is. But as long as the ladies think it's sexy, those guys will get to breed, but if they're not able to run from predators, they might get eaten before they can breed. So there's a fine line there. You go over it, you're going to be eaten. You're too small, you're not going to breed. So there's definitely too big, I'm dinner, too little, I don't get to breed, stabilizing selection. But it's sexual selection. So again, even if it's sexual, artificial, natural, all of these guys have these directional, disruptive, or stabilizing types of selection that's going on within them. Okay? Does that make sense? All right. Mutations. What are these guys good for?
Diversity.
Genetic diversity. So they're very, very important for what's our smallest unit that can evolve?
Population.
Populations, yes. I know, you got that one wrong, you'll never forget now. So it is good for the population, but it tends to suck for who? The individual. Yeah, a lot of her mutations tend to be not good, maladaptive. Every once in a while you get something that's super useful, and then sometimes you get a mutation that does nothing. So in terms of coding for a protein, it may code for the exact same amino acid, right? That was what we talked about when we have more than one code for an amino acid. What's that called? Oh, I switched gears on you.
More than one code.
That's multiple choice, so always I see it.
So it starts with an R. Redundancy, yeah. So that redundancy in the code, so you could have a mutation you pass on to your offspring during genetic recombination, or you have a little blurb that happens because you kept your cell phone next to your gametes too often. But it didn't change anything. It coded for the same amino acid. You guys do know cell phones give off radiation, right? Okay. Yes, don't keep them by your gametes, fetuses, any of that. All right. So as long as it codes for the same amino acid, it's a neutral mutation. It's a mutation, but it doesn't hurt anything. Okay? It doesn't help anything. It's just neutral. All righty. Good on that one. Evolution's pretty straightforward on that. 20. These guys, again, this tends to be also pretty straightforward. Again, the difference, what's anatomy? Structure, function? Structure. And then physiology, function. What are your 4 basic tissue types?
Epithelial.
Connective?
Muscular.
Muscle? Epithelial?
Nervous.
And your acronym?
Semen.
Semen. Yeah, C-M-E-N. All right, basic function and your basic organs. I mean, if we ask you, the stomach, which system does that belong to? You should know digestive, yes. Homeostasis, negative feedback, positive feedback. All right? Are we feeling comfortable about those, for the most part? What are our 2 examples of positive feedback only?
Blood clot and babies.
Yes, blood
Labor.
clotting and labor, okay? Everything else is negative. Even if it's doing something that's great for the body, it's a negative feedback system. With a negative, do we enhance the stimulus or reverse the stimulus? Reverse the stimulus, yes. So blood sugar regulation, both up and down,
Negative.
is a negative feedback system for both. Yes?
So isn't it the need to go to the bathroom a part of this?
No, because you're trying to reverse the stimulus, which is the stretch receptors. Your brain is signaling that the bladder, the muscle is stretching, and you want to reverse that stimulus and let the muscle contract.
So that's a negative. So, let's just say that our teacher said it was positive. What is the test going to say?
It's negative.
Okay.
Yeah, there's really only two. Blood clotting and labor are positive.
Okay, and everything else is
Everything else is negative. It's easier to remember that than to try to remember what's negative. Yeah. I know, sometimes they seem like they're positive, but you gotta look at what the stimulus is. And that's that stretch. Now, eventually, there's gonna be pain involved with that as well. And we're gonna try to reverse that as well. But full bladders, pain, agony.
Right.
All right. Good on those? All right. So, big picture for the first part of this then. When we talk about this, okay? So again, looking back at those themes that we've been running through, look back at how current information ties back to earlier chapters. So like I say, we talked about all of those monomers, polymers, dehydration synthesis. We talked about hydrolysis. All these things come into play here. Do you guys want to take a little break? Is that, no? Keep going. You're not eating any food. Everybody brought some snacks. Okay. All right. So take a look at all the big stuff, like I say, as we've been looking at these guys, the overall pictures that we keep talking to you about over and over and over. You know, what are we asking you about? What are we reiterating? All this kind of stuff. So definitely the hierarchies, they'll be in there. Make sure that if we gave you, like the stomach, would you be able to classify it within the hierarchy? Is it a tissue? Is it a cell? Is it an organ system? Is it an organ? It's an organ. Yes. Okay. Yeah. So make sure that you're comfortable being able to do that. Back to our bonds. Ionic, hydrogen, covalent. Which is our weakest within those three? Hydrogen. Are there any electrons involved in hydrogen bonding? Any exchange or movement of electrons? You asking me or telling me? They're weakest, so there are no electrons involved in terms of exchange, sharing, or anything else. Usually we're looking at a molecule having an unequal distribution of electrons. That creates positive and negative signs, right? Usually involving a hydrogen who's lost his little electron. That's why we call it.
Bullet.
Yeah, the hydrogen bond. Yeah, okay. Ionic bonding, how does that work?
It is unequal sharing. It's like tugging.
Is it sharing, or is it just absolutely giving away or terribly taking?
It's giving away, gaining or losing.
Gain or lose. Yeah, there's no sharing involved here. It's all or nothing.
And which one was that?
This is ionic. Okay.
Sharon?
Ionic is not sharing. Yes, ionic is I get them all. They all belong to me. I get them. That's how I remember it. Okay. So ionic is gain or lose, no sharing. But what's going to happen to the charge on an atom if you gain or lose electrons? If you gain electrons, it becomes negative. If you give electrons away, the charge becomes positive. And then what do we know about positive and negative ions? Attraction, because opposites attract. Yes. So we get attraction, and that's how our ionic bonds are formed. How easy are those to get apart? Fairly easy. I mean, what will pull them apart? What substance? Water. Water can rip ionic bonds to shreds. All right, let's talk about covalent, cohabitating, sharing. So you're sharing bond, or electrons through a bond, but we have two types of sharing.
Polar and non-polar.
Polar and nonpolar. Which one is nice? And carbon does this, and it's equal distribution of the electrons. Nonpolar. So anything with carbon shares nicely, and we get equal distribution of electrons. Do we get any hydrogen bonding with that? Are you asking or telling me?
Anything with carbon, you said.
Anything with carbon,
We do, because there's hydrocarbons.
It's non-polar. so there's no hydrogen bonding with these guys.
It is covalent all carbon.
No, well, covalent all carbon? No, not at all. Oxygen and hydrogen for water, that's gonna be covalently bonded, and that's gonna be our polar example
Okay.
for oxygen hot. So I always say carbon shares nicely, so when carbon is involved, it's a love fest and there's always equal sharing.
Okay.
But if oxygen's involved, with certainly, especially with hydrogen, it's not gonna share nicely. Okay?
Okay.
However, carbon and oxygen can duke it out, and carbon will make oxygen play nice. This is coming from a biologist, not a chemist, so they would cringe if they heard me talking about it like that, but that's how I learned it.
So and then hydrogen.
Hydrogen just doesn't have a lot of pull, so if oxygen is coming along and it's got more what we say electronegativity, it hogs, it can pull the electron right away from hydrogen. Now that creates a strong bond, a covalent bond, but it creates a molecule that is bent and got charges, partial charges, what is that called? Polar. It's a polar molecule, partial charges on it. Right? So when we look at water, make sure you know your water again, guys. Water sticks to its health, its properties, how that plays a role in the body, how it clings to not only itself and other things. Okay? Water is going to be big, and it's huge. It's huge in anatomy and physiology. Okay? So really, really, not just for the test, make sure you understand water before you move on. All right? pH, what is that a measure of? Yes, the silly scale. Potential hydrogen of a negative logarithmic scale, so high numbers actually mean basic and low numbers of which ion? Hydrogen. Yeah. So very high numbers actually mean low numbers of hydrogen, and very low numbers
Very high.
mean hydrogen numbers are high. So very low numbers mean really acidic. Really high numbers mean
Basic.
very basic. What does 7 mean? Neutral. What does that really mean in terms of numbers of hydrogen and hydroxyls? Equal numbers, right? So neutral means equal numbers of each. When we talk about a buffer system in the blood, the bicarbonate buffer system, that was in one of your homeostasis packets. What does a buffering system do?
It maintains.
Stabilize one condition,
What kind of condition?
whatever it's programmed to do.
I think you're on the right track. Are you talking about pH?
Yeah,
Yes.
whatever,
Okay.
whatever one it's meant to do, whether.
Okay, so it can, yes, so it's gonna stabilize pH. Yep, buffering systems stabilize pH. Some directions better than others. Yeah, the bicarbonate buffering system was better at stabilizing for acidic or basic conditions. Remember? It's okay if you don't, do not panic, this is not on the test, okay?
I think basic.
I think it's
Acidic, yeah, because we tend to run more acidic. What happens when you put carbon dioxide in water? What do you create? An acid, carbonic acid. Yeah, and our blood is mostly
carbon.
water, and it's carrying carbon dioxide, so the more you run around, the more things you do, the more you do in metabolism, the more carbon dioxide building up in your bloodstream, and your blood is sliding towards acidity. Yes. So our buffer holds it. Okay, can you overwhelm a buffer? Yeah.
Eventually.
Yeah, absolutely. You can overwhelm a buffer and cause it, the pH to crash or to shoot way up, depending upon which way you're going. Yes, you can. All right. Macromolecules, what are the 4 big ones? Carbohydrates, proteins, lipids, and nucleic acids. All right? What are the monomers of carbohydrates? Those are the specific ones.
Saccharide.
Generic name.
Monosaccharide.
Oh, you got We have saccharides, right, monosaccharides, polysaccharides, and disaccharides, okay. So when we talk about the actual really single building block, it's a
Monosaccharide.
monosaccharide. If we put two of them together, like sucrose sugar,
Yes, a disaccharide.
disaccharide, we put a lot of them together, like starch or glycogen, polysaccharides, yes. All right. How are they joined together? What type of bonds? Don't make it too hard. This is ionic, covalent, or hydrogen. Carbon's involved. Covalent, yes. All right. What do we say next? Proteins. What are your monomers for proteins? Another name for amino acids.
Peptide.
Peptides. Okay, don't forget another name for amino acids is peptides. What type of bonds form? Peptide bonds, yeah. So this is the one that we actually make you know. What kind of bond is a peptide bond? Ionic, covalent, hydrogen. You sure? Bet you're great on it, Jesse?
Yes.
Excellent. She's right, they are covalent bonds. Alrighty. How do we break a protein apart? When you have pepsin in your stomach, it's an enzyme. What is it? Hydrolysis. Is water needed or is water created for
Water
hydrolysis?
is needed. It's hydrolysis, so it's
Hydrolysis,
water
so
breaking. We need water to
we
break.
need water. We have to split it. Why do we have to split it? What would happen to those monomers if we didn't put a hydrogen and a hydroxyl group back on them? They're going to bond to something else. Yep, they're going to be reactive. So we've got to neutralize them by splitting the water and putting it on. All right, lipids. These guys are kind of unusual. They don't run in big long chains like our other types of polymers.
They're a fatty acid and a triglyceride.
No, you're careful.
Oh, glycerol.
A glycerol. There you go. Yeah, and they have the two different types of monomers,
So they have
the
a
fatty acid
fatty acid.
and the glycerol.
And then they have a
If we have how many fatty acids on a glycerol?
3
3. It becomes a
triglyceride.
triglyceride. All right, if we have a double bond in the tail of the fatty acid, is that a saturated or unsaturated fat?
Unsaturated.
Excellent, unsaturated. All right. Is that a covalent bond that holds the fatty acid to the glycerol, an ionic bond, or a hydrogen bond? Good one, Sam. All right. Don't forget, we put all of these together the same way. No matter what polymer it is, we take them all apart the same way. Absolutely identical. We put them all together through what process? Dehydration synthesis. We take them all apart through what process?
Hydrolysis.
And every bond that we break is the same. It's a covalent bond. Okay? So don't forget, this is all the same thing. We're just changing out the pieces. All right. Our last one, nucleic acids. Monomers? Nucleotides. What are the 3 parts to a nucleotide? Sugar, phosphate, and nitrogenous base. DNA has how many different base types? And RNA has how many different base types?
4.
How many do they have in common?
3.
Very good. Ooh, spunky. You know your DNA. All right. Comfortable with that?
What are the macromolecules again?
Proteins, lipids, carbohydrates, nucleic acids. And truly, truly of those 4, which is really the largest?
Carbohydrates.
Which ones did you guys see in the onion root tips? With the microscopes at 40x, you could see this molecule. They were in different stages, prophase, metaphase, anaphase. Nucleic acids, chromosomes, guys. Right? They were huge. You could see them with just 400x magnification. That's amazing. Yes, nucleic acids are really big. All right. Back to your cells, all your organelles again, all of your functions. Make sure you know these, okay? This is important, like I say. When we move on to anatomy and physiology, we'll talk about how cells are modified from the basics, so you have to know what the basic is. All right. Passive, active, transport. Which one's going with the gradient?
Passive.
Passive, okay? Active transport,
Against the gradient.
against, is that always true?
Most of it's always true, right?
There's always exceptions.
There's always exceptions, not for passive. Passive is always with the gradient. When would you need active transport, but you're going with the gradient? What was it? Yes, very good. Really big molecules where they're not going to fit through. So what's the process of moving a humongous molecule out in a vesicle? You move it, vesicle up to the wall. What's that process called? Moving it out of the cell.
Exocytosis.
Exocytosis. There we go. Exiting the cell. Exocytosis. Same thing if you want to move a big piece of something into the cell. That process is
Endocytosis.
endocytosis. It can be with the gradient, but is that active or passive? Active. Absolutely active. Okay? So there are some cases where we are moving with the gradient, but it is active. Passive is always with the gradient, is always with, and no energy being involved. All right. Osmosis is the movement of what?
Water.
Water where?
Towards the solute.
Across a
Gradient.
membrane. Remember that osmosis is movement of water across a membrane. Otherwise, if water is just moving around, it's diffusion. Yes. Okay? So water can diffuse, air diffuses, but if you move water across a membrane, then we're talking osmosis. Hypertonic, hypotonic, isotonic. What do you remember about those? Okay, so tonic referring to
The solute level.
solute concentration in the solution, yeah. Iso means that,
Happy.
yeah, whenever we're talking about iso, hypo, hypertonic, we're always talking about comparison of two solutions, at least two, right? So iso means the solutions are equal in solute levels. Which direction would the water flow?
Oh, it go in and out as it's needed.
There we go. It would just flow in and out at equal rates. Please never say it wouldn't go anywhere, okay? It always moves. That one, they will nail your butt to the wall, NA&P on that one, okay? It's always moving, but there would be no net movement. But there would be movement. All right. Hypotonic solutions, do they have more solute or less solute? Hypo? Less, okay. Then the solution they're being compared to would be considered hyper, okay. If one solution is hypo, the other one has to be hyper. All right,
So hypo is less concentration of?
less solution, less solute, let's put it that way, less solute,
Okay.
okay, than the other solution. All right. So in a hypertonic, I'm sorry, go ahead and ask. Okay, give it a shot if I don't ask. In a hypertonic solution, sitting next to a hypotonic solution with a semi-permeable membrane for the movement of water but not solute, will water move into the hypotonic solution or will water move into the hypertonic solution?
Hyper.
Less less water, less solute, less solution, it's drier over there. So water's gonna flow uphill against this gradient.
Yes. It's gonna move towards the solute.
It's gonna move towards the solute. So there's less water in the hypertonic, so there'd actually be lower, so there's more water in the hypo. So it moved down its gradient into the hypertonic solution.
Okay.
Or, the other way that I do it sounds like Kim does too, water always follows solute, okay? But you gotta keep track of it. Either water's flowing down its concentration gradient, so the hypotonic solution would have more water than the hypertonic solution. So water would move down its gradient into the hypertonic solution. Or, the easier way for me, whatever works for you, wherever the solute's higher, water follows it. Okay? Okay. So if I have a kidney tubule, okay, it has a high concentration of water in it, low concentration out here, the water will move out. Does that make sense?
So that's a permeable membrane.
That's a permeable membrane.
Okay.
To keep this concentration where I need it, I actively pump, let's make it correct, salt out into this area. That means the salt concentration is high out here. Okay? So, would I want to actively pump out salt into that kidney if I had just drank 2 gallons of water? Wow, this got just a whole lot more complicated than you expected. Do you want water to leave the filtrate? Anything you don't pull out is going to become urine. You just drank 2 gallons of water. Do you want to move salt out of the filtrate, or do you want to leave salt in the filtrate?
Oh, we need
Okay, back it up before you panic. Ask yourself, do I want to get rid of water, or do I want to keep all of those 2 gallons in my body? Okay, I want to get rid of the water, so I want the water to stay in the filtrate. Where do I want those salt to go then?
in the
To
filter.
stay in the filtrate or leave? In the filtrate. So I want to leave salt in the filtrate to keep the water in there with it, right? Does that make sense? Okay. That's where you're going to go with hypertonic, hypotonic, and the flow of water and solutes. Okay, and then you'll talk about all the hormones that regulate salt in or out to regulate water. Okay, so this is a biggie. All right. Energy, endergonic, exergonic. Protein synthesis in the ribosome, is that an endergonic or an exergonic process?
It's taking energy, so it's extra it's taking.
So you're making bonds or breaking bonds? If you're making bonds, it's endergonic or exergonic? Endergonic. Energy in. To build a bond takes energy. To break a bond releases energy. So protein synthesis, is it endergonic or exergonic? Endergonic. Definitely endergonic. Yep. Okay. Hydrolysis of protein in your stomach. You've just eaten a Big Mac. Okay, we're going to guess that it's actually protein in there. Carbohydrates, cheese. Okay, kind of cheese. that an exergonic or an endergonic process? Exergonic. The energy needed to get those things started, though, what is that called? Activation energy, right? And who is running those shows or breaking things down? What proteins, mostly? Starts with an E. Enzymes, right? So don't forget your enzymes and how they work. Need an energy of activation, but they reduce the overall input, make it much cheaper. All right. What type of bonds hold enzymes together if they're proteins? Covalent. Holds what level together? Primary, secondary, tertiary? And what are peptide bonds? Are they ionic, hydrogen, or covalent? Yes. So the primary level is held together by peptide or covalent bonds. How do I give it its 3D shape? Is what? What kind of bonds give it its 3D shape? For the most part.
Hydrogen bond.
Our weakest ones? Hydrogen. Yeah. Right? So we're looking at our amino acids that have positive, negative polarity on them, being attracted to each other, causing folding and hydrogen bonding. Sound right? Vaguely familiar?
The primary hold is the covalent bond.
Yep. Yep. A string of amino acids. All right. What types of things then will affect hydrogen bonding?
Salt concentrations and
Okay, so salt concentrations,
temperature,
temperature.
pH.
Okay, temperature in what way? Hot or cold? Hot. Too hot and they will
Denature.
denature. But what happens to enzyme activity as we warm things up? Does it go up or down? Why does it go up?
There's more molecular movement. There's more bouncing around, more molecular movement.
Okay, so we talk about the enzyme. What does it work on? What's the official name for the molecule it works on? Substrate. Yes. So how do the enzyme and the substrate find each other?
Bouncing around.
Bounce around. So the more faster they bounce, the faster they find each other, right? How does cold work on enzymes? Does it break the hydrogen bonds? No, is correct. But it does what to the kinetic energy of the system? Decreases it, right? So they don't bump into each other as often. All right, so salt concentrations, any ionic concentrations, so when we talk about that, ions can do that. That also brings us then to pH. Can break that positive, negative bonding, cause those problems. You guys are familiar with that, right? Vaguely. Okay. So being able to, big pictures here, compare and contrast aerobic versus anaerobic definitions. We're not going to ask you the teeny tiny details we asked you on the midterm about these guys, but you should know the big pictures at this point. What's going on with oxidation? What's reduction?
Oil rig.
Oil rig. Oxidation is loss of electrons. Reduction is gain. Yes. What are those two reactions called when we put them together?
Oxidative.
Redox.
Redox.
Redox reactions. There you go.
I was getting
That
into
will
something
lead
oxidative.
to the electron transport chain where we do, or I guess it would be actually the Krebs cycle where we do oxidative phosphorylation. Yeah, in the electron transport chain. All right. So big things on this case, again, just where they occur, starting reactants, products, what you get, ATP amounts for each part. We're not going to come back and ask you the teeny tiny details, you know, on all of that. But the big picture here, again, is really important on this. All right. Do you guys have any questions for me? Anything else you want to explain?
So how many APPs produced? Can you go over them again?
Yeah, you want to go over aerobic versus anaerobic?
Yeah, please.
Absolutely. Okay. So what is common between aerobic and anaerobic process? What's exactly the same?
Is the citric acid cycle, is that the same?
Nope, it's the first part. Glycolysis. The splitting of the sugar. Exactly the same no matter which one you do. So how many ATP are invested to get that to start?
2.
2. A gain of, slow down.
A gain of 2,
A
so
gain of glycolysis.
4.
A net gain of 2, but a total of
4.
4. Okay. What do we have when we end glycolysis?
Pyruvate.
Pyruvate. All right. So the question here now becomes, this is where anaerobic versus aerobic diverge. If we're doing anaerobic, pyruvate is going to be changed into what?
Acetylcholine.
Lactic acid. Yeah.
Okay.
And recycled back through eventually through the liver. We don't get much more gain out of that. Okay? If it's aerobic respiration, where is it going next?
To the
Citric acid cycle. Yep. And what are we going to do here?
attach acetylcholine.
Do what?
We're gonna put a tie on him before he goes into
He's
the
gonna
club.
go into the strip
Acetylcholine.
club, so we put a tie on him. Where we do what? What are we stripping? Electrons, right? So we're harvesting electrons off of this molecule and loading them onto
The electron transport chain.
transport chains, and the shuttles are taking them there. All right? How many ATP per glucose are produced in the citric acid cycle?
2.
2. 1 per pyruvate, but there's 2 pyruvates per glucose. Our big gain, though, is the electrons. Right?
Mhm.
What is the job of the electrons? Once we take them to the ETC, or the electron transport chain.
They're gonna build a gradient where the hydrogen goes
Yes.
out.
So they become the energy source for our pumping stations. Right? And those pumping stations are going to move hydrogen ions
Out of the
out of the matrix area into the intermembrane space, create a big hydrogen ion gradient. That's what electrons are doing. How do we get them off the electron transport chain?
oxygen.
Oxygen sits at the bottom and pulls them off, because we know oxygen loves electrons. So it hogs them, pulls them off. But now oxygen is unbalanced, so what do we have to do to balance it?
Make it into water.
Make it into water. So we add a couple of hydrogen ions, and it becomes water. How many water are produced in this entire process?
6.
6. Okay? How many oxygen are needed to run this process? How many glucose? One glucose, 6 oxygen, 6 water, and how many CO2s will be produced?
12.
6. Yeah, 1, then 6, 6, 6 is going to be, I know, very scary. Or a good year for Queen Elizabeth, or whatever it was, like 1666. Anyway. But yes, okay, so we're going to produce carbon dioxides. Where are the most carbon dioxides produced? Glycolysis or in the Krebs cycle, citric acid cycle?
Citric acid cycle.
Yep, Krebs cycle. All right. How do we actually make the ATP then in the last part of this whole process? Big gradient of protons. Phosphorylation, yes. And we use what enzyme?
The synthase.
Yes, what kind of synthase to make ATP? ATP synthase. There you go. That's why I was throwing at you. It was too easy. You didn't trust yourself. Yes, and so as the protons shoot back through, going down their gradient, they spin the enzyme, create ATP. How many?
42.
Yeah, 32 to 34. So we end up with a net gain of 36 to 38 from one glucose. 32? At that spot, I think you're right, yeah, 32. And then it's, yep, I'm sorry, yep, it's the 2 out of the glycolysis and the 2 at the Krebs cycle. Yes. I lied, yeah. I did that wrong. All right, anything else? You guys
Were
good?
we gonna get our papers, some of our papers back today?
You were. Yesterday ended up being way more involved than I planned, so I don't have anything more dated for you than what I have right now. So,
As far as the final, there you said that there's going to be 2 essay questions.
And those are 5 pointers each.
yep,
Do we know which those gonna be on? Which? We don't.
it can be old stuff, it can be new stuff, it can be the whole thing, but old stuff would be big concept kind of things, you know? The new stuff would be the detail.
Would it be one of those where you have to draw a chart, something like that, where you, maybe?
Maybe. Charts are always good. I mean, you know, I have a variety of things to pick
Like a
from.
homeostasis chart, where you got one of those, or?
Probably not I mean, not like, the packet material from homeostasis packets is not on the test.
Okay.
If that was what you were thinking, yes, it is not. But things like Punnett squares, the varied and contrasting meiosis, mitosis, describing protein synthesis, all of those big concepts just scream essay questions.
Okay.
Okay?